This diagram shows the general life cycle of a fungus, and it's really quite different from our life cycle, so I want to go over it so that you're familiar with it. One of the strange things about the fungal life cycle is that for most of the time the organism is haploid. That is, it only has one copy of the genome in each of its cells. We're used to diploid cells, where the cell has a copy of the genome that inherited from mom and one that inherited from dad. The fungal life cycle is flipped around from what we think of as normal. In our bodies, we are diploid almost all the time, and there's a very brief life cycle stage called the gamete that is haploid. With fungi, it's reversed. Most of the time they're haploid, and there's a very brief stage called the zygote that is diploid. In addition, there's a third kind of ploidy that you see in the fungal life cycle. This is called these parts that you see in brown are parts of the life cycle where the fungus is heterokaryotic. That is a peculiar state that never happens in the human life cycle, and I'll go over it in just a minute. As I mentioned, the body of a fungus that you usually see is the mycelium, and it is haploid. All of these cells have one copy of the genome. And if they have two, it's just because one nucleus floated over from another cell. These mycelia can produce spores asexually. What happens is that mitotically, this mycelium grows a spore-producing structure, just like all the cells in the mycelium. These are haploid, and so are the spores that are made. Those spores, if they land on a place where there's food, will grow into a mycelium of their own, but through mitosis. So there's no genetic change going on. You've basically got a haploid mycelium giving rise to more haploid mycelia that are genetically identical to itself. However, fungi also can generate spores through sexual reproduction, and that's this other cycle over here. What happens in this case is that fungi, fungal hyphae rather, from two different individuals meet and fuse. What's strange about this is that the nuclei of those two individuals do not fuse. So here's one individual up here. It's genetically different than this one up here, and the artist has chosen to represent that by having nuclei that are one color down here and nuclei that are another color up here. When these hyphae meet, and they're of different mating types, that's usually how it's determined in different groups of fungi. When they meet, these cells fuse, but the nuclei don't. Instead, what you get is a new mycelium where each cell contains two nuclei, one from each parent. And those nuclei will go ahead and divide and grow independently of one another. This is called plasmogamy, is the fusion of the hyphae from the two different individuals. And what it yields is a heterokaryotic mycelium, neither haploid nor diploid. Instead, it's sort of haploid plus haploid. It's got two haploid nuclei hanging out in each cell, behaving more or less independently. Eventually, what this mycelium does is it generates a zygote. And it's in that zygote that the nuclei actually fuse so that you have a diploid cell. And that diploid zygote then undergoes meiosis. That meiosis results in haploid spore-producing structure and haploid spores. Now, one thing to note, though, is that even though these spores and these over here are both haploid, these are all genetically different from one another. So it's in this part of the life cycle that fungi generate individuals that are genetically unique and novel. So how is it that fungi find their food and grow and reproduce? Well, as I mentioned earlier, they are very, very good at absorbing things. The key thing that fungi do is they absorb things from their environment. They can do this in a couple different ways. They can absorb high energy compounds from dead matter. Usually what they have to do is decompose or break down dead organic matter so that they can harvest the usable high energy compounds. But there are also fungi that do the same thing within living organisms. It's a pretty small step between breaking down dead organic matter and eating it to breaking down live organic matter and eating it. And accordingly, this transition has been made many times within the evolutionary history of the fungi. As I mentioned, it's the high surface area to volume ratio that is key to fungi being good absorbers. But they have a few other traits that make them very, very good decomposers. One is that they use extracellular digestion. You should remember from the last lecture when we were talking about protists that lacking the cell wall and having that dynamic cytoskeleton allows some eukaryotic protists to basically change the shape of their cells so they engulf food. They can bring in large particles of food. Fungi, however, because they have a cell wall, can't do this. Very large molecules are too large to be brought in through the cell wall and the cell membrane. So what fungi have to do is actually digest them outside of the cell, break them down into smaller molecules that can be absorbed through the cell membrane. So for example, starch and cellulose can be broken down into monosaccharides that can be drawn into the cell. But in order to do this, fungi have to secrete digestive enzymes that move outside of their body and digest those chemical compounds outside of the fungus while they're still basically out in the environment. Then, of course, those compounds can be absorbed by the hyphae and used to grow the fungus. One other trait that some fungi have, not all, is the ability to break down lignin. This is unique to the Basidiomycetes, which is a group of fungi that we'll be talking about in a little more detail later. These are the only organisms on Earth that can break down lignin. Lignin is the very hard polymer, or actually a family of polymers, that makes up wood. It's what makes wood tough and woody. As you can see here, these chemical compounds can be extremely complicated. And what makes them really tricky to break down is there are all sorts of different linkages in here. There's a tremendous diversity of ways that these subunits can be connected to one another. And typically an enzyme only targets, one or two different linkages. Lignin is very difficult to break down because it consists of an almost infinite variety of these linkages. And you would have to have an almost unlimited diversity of enzymes to break it all apart. However, the Basidiomycetes have discovered a trick that can degrade lignin completely into carbon dioxide. As it happens, this is very useful to you because the making of soft, pliable things like paper products like toilet paper is only possible because you can treat wood with fungi that takes out that woody tissue. If we didn't have this, all of your toilet paper would contain woody tissue, and you would be sorry about it. What the Basidiomycetes do is actually a pretty clever trick. They secrete an enzyme outside of their bodies that strips away a single electron from a molecule of lignin. The removal of this electron leaves what is called a free radical. Free radicals are extremely reactive, and if they get inside your body, they're quite dangerous. Because what that free radical will do is to correct the imbalance that it now has electrically. It will strip an electron away from a different molecule. Now that molecule is a free radical, and it will in turn strip away an electron from some other molecule, and so on. It's this chain reaction of electrons moving around. And at each movement of an electron, you can damage or break apart the chemical compound that's being affected. Basically, what the fungus is doing is it's lighting a match to one part of the lignin molecule. And what happens is essentially enzymatic combustion. It's a little bit like lighting gasoline on fire. Um, in great contrast to most enzymatic reactions, this is really uncontrolled. What happens is that the lignin molecules just start coming apart all over the place until finally all that you're left, they become more and more heavily oxidized as the process goes along, until all you're left with are the most oxidized possible carbon compounds, that is CO2. In this way, the Basidiomycete gets around the fact that all of these linkages within the lignin molecule are different. It's using a chemical reaction that can rip apart all of those linkages indiscriminately. One of the shortcomings of this method of breaking down lignin, however, is that partly because this is totally uncontrolled and partly because it's happening outside of the cell, the Basidiomycete can't capture any of the energy that's released in these oxidation reactions. The reason that these fungi have evolved to do this at all is that within dead plant material, there is stuff, food stuff, that they can break down and collect high energy carbon compounds from. Typically, this is cellulose. Even cellulose is tough to digest, but there are a few organisms on Earth that can break cellulose down into monosaccharides or disaccharides that can be used for energy.